Okay, for our next talk, we'll stay in India, and Dr. Jane Carlton will talk about the India isomer.
Is that good? Great, thank you. Well, thanks very much for the invitation, Peter. I think this is a fantastic meeting. We should have one every year or every other year. Anyway, it would be nice to see us all coming together regularly for this. I'm going to be talking about the Center for the Study of Complex Malaria in India. This is the group of investigators here. Let's see if I can get the mouse. Oh, the mouse is going to be here. That's right. So this is a true partnership. There's a level of sophistication in terms of the research which is undertaken in India, which means that our collaborators are true partners in India. And we have collaborated with the National Institute of Malaria Research, shown here. This is many of us here at New York University, at Penn State, and then also at SAIL down here. And let me tell you a little bit about the National Institute for Malaria Research, which is where our collaborators are based. Under the directorship of Dr. Nina Valecha, shown here, this is the headquarters in Delhi, a purpose-built building with insectaries and lab facilities that was built about 8 years ago. And NIMR is the only institute in India which does basic and applied research. It's very well connected to the National Vector-Borne Disease Control Program as well. And they run these 10 or 12 field sites, as you can see roundabout in India here. And we actually have partnered up with 3 of those field units that I'll be telling you a little bit about. Down here, this is SAIL. So this is the a new component, actually. So this is Sanjeev Mahanti and Dr. Satapathy as well. And this is at Ispat General Hospital. And this is one of the hospitals in India in an endemic region which has an MRI machine. And we have partnered up with them and with colleagues in Malawi, and I'll be telling you a lot about that work as well. So I don't need to give you too much background about India and malaria because Pradeep did a lot of that for me. But basically, this is a map from 2000. We know that malaria has decreased in incidence a lot since 2000 in the past 15, 16 years. This is the real hotbed here. So this is the central eastern states, and then also up here, the eastern states too. Falciparum and vivax are the predominant species. There's some P. malariae that we get in one of our field sites. 6 Anopheles species are thought to transmit malaria. And this is really how the malaria situation has decreased over the past decade or so. These are figures taken from the National Vector-Borne Disease Control Program. You can see probably wildly inaccurate. That's about 400 deaths from falciparum, which we really know is inaccurate. But the total number of cases really decreasing over that period. And as I said, a number of Anopheles species that transmit the parasite, very complicated species complexes, but these are located in what is thought to be different regions of India. So Stevens eye is the main urban vector. Culissifaces, which causes about 60% of the malaria, is found in rural and peri-urban areas too. So our center is called, as I said, the Center for the Study of Complex Malaria in India. And why did we call it that? Well, it's because malaria infections in India are highly complex. An individual can be infected with more than one Plasmodium species, vivax or falciparum, and occasionally malariae. And an individual can also be infected with more than one genetically different strain as well. And so why does this matter? Well, for a whole host of reasons. Infection dynamics influences clinical outcome. If you're infected with falciparum, you're going to go on to develop cerebral malaria, for example. If you're infected with vivax, will you have the hypnozoite formed and potential for relapses? Mixed genotypes enable generation of novel strains, of course, and competition between clones within a host selects for increased resistance and virulence. So our overall theme is the analysis of single and mixed species and single and mixed genotype infections, which we call together complex malaria, generated by falciparum and vivax, and how this influences disease outcome, vector transmission, and drug resistance. And our three projects, which were NIAID mandated projects, we have an ongoing epidemiology project to determine the baseline epidemiology, genetic diversity at 3 different sites, a transmission project run by colleagues at Penn State, really looking at the environmental conditions that determine malaria transmission at 2 of our sites, and then a large genomics project, developing a next-generation sequencing core to look at genetic diversity in vivax, and then also track the changes in frequency of clones within a patient before and after drug treatment. And just to show you our field units, I wanted to show you this quick movie. This is India, and our field sites should be coming up here. So we're going to zoom in to Rourkela, and I hope nobody gets motion sickness from this. This is our forested area. Anopheles culicifacies is the major vector. You can really see how forested it is. Plasmodium falciparum is the major species, Plasmodium vivax less so. And we work in a number of these villages, as you can see here. Our field teams go out, enroll patients, and collect samples. And the main town is called Rourkela. As we come out, we'll go up, or down the coast, I should say, to Chennai. So this is our urban malaria site. This is our field station here that NIMR has. And now we're zooming in then to, in particular, the Besant Nagar catchment area. So these are areas along the coast, this is the sea here, where there are slum houses, fishermen's houses. And these houses in particular, these places, are hotspots for a lot of Plasmodium transmission. This is Plasmodium vivax, by the way, and no. Okay, Plasmodium vivax. And Anopheles stevensii is the major vector. That was that was Nadiad, where there's a mixture of vivax and falciparum, and culicifacies is the majority vector there, and that was a peri-urban sort of deforested area. So those are our 3 field units. Okay. So we spent the first few years of the project renovating and equipping our 3 field study sites. Maybe one of the things I should mention, Pradeep didn't mention, but we have to have Government of India approval before we can work in India, and that took approximately a year. So in fact, our field sites have not been going for, they've only been going for about 4.5 years now. And a lot of the time we were taking known field stations, which NIMR had developed, renovating them and updating them with equipment. This is a visit that Mala took to our particular unit when we had an opening by the CEO of the Rourkela steel plant. Lots of fanfare. We also undertook staffing and training and technology transfer as well in Rourkela, Chennai, and Nadiad. These are some of the photographs here. This is us setting up molecular biology labs at each of the 3 sites. This is some training going on for Anopheles larvae identification. This is the sense of relief on the data entry operator when she found hanging folders, finally, for the filing cabinet to put the consent forms in. And at each of the 3 sites, then, we have undertaken these epidemiology projects. And the schematic is shown here. So we start each of the projects with a census at the 3 sites, and then we undertake passive and active epidemiology studies. So for the passive surveillance, it's a clinic study. These are people coming into clinics at the 3 different sites where they're screened and enrolled and then followed up to about day 42. I must say day 42 is probably not long enough for many of the studies involving Vivax. We didn't get very many relapses in that time. So going forward, I think we might change that. Then for the cross-sectional and longitudinal studies, shown here, so the cross-sectional was 400 subjects at random 4 times a year, and the longitudinal was 100 subjects, 100 houses, I should say, followed 4 times a year. We also undertook a reactive case detection project as well, which was just done at a couple of sites. I'm not going to go through all of these because there's too much. I'm going to pick a few key projects with interesting findings. And this is just to show as well that for each of the samples, for each individual, the samples were collected, which were blood spots, slides, microvet, and RDT. And these were then processed. If positive, Vacutainer was collected, RNA, DNA, species-specific PCR. Oops. And then these would go on to be genotyped by microsatellites, single-copy genes, amplicon sequencing, whole genome sequencing. The sera was used for ELISAs and the Feldner protein arrays. I won't have time to talk about those. And then also for transmission studies, too. So we've been doing a lot of those. Transmission at NIMR, particularly in Chennai, is quite routine now. And then all of the samples and all of the data entered into our REDCap database. So some of the findings. Well, our first question was, what is the baseline epidemiology of malaria at our 3 field sites? And these were some of the people involved at in Nadiad, Rourkela, and Chennai, what are called the officers in charge at NIMR, and then also at NYU, our epidemiologist, Anna Maria van Eyck. What we did find, looking at all of the data so far, is that there's a high burden of asymptomatic and submicroscopic infections. This is actually the first time that PCR detection of Plasmodium has happened in India at multiple sites. There have only been a couple of studies previously on a much smaller number of samples. We found malaria prevalence very low at all 3 sites. You can see by PCR indicated here. Vivax dominating, as we had previously thought, a ratio of 70 to 30 in these 2 sites. Falciparum predominating in Rourkela, 80 to 20. Almost all of our Vivax infections have gametocytes, very similar to what Joe was saying. Risk factors for malaria were being male in the season, significant anemia, and we really see a high burden of asymptomatic infections. Up to 73% of our detected infections were asymptomatic. And our submicroscopic infections was also high, up to 70%. And that's shown in this graph here. So if you just take a look at the microscopic infections, so falciparum being very dominant in Rourkela, Vivax in Chennai, then these are the submicroscopic burden you can see here. We're really missing a lot due to those submicroscopic infections. And I think this really gives us an idea of going forward now that India has announced its elimination plan of what to focus on. Some other interesting findings from our census. We had a look at the common use of mosquito repellents. So in India in particular, and I think in China too, there's an extensive market for mosquito repellents such as mats and coils. You've probably all seen these. That's a coil. This requires electricity. It's a vaporizer. They constitute about a 1.5 billion industry, which makes up about close to 3% of the per capita income per year. So it's really quite an industry. There are, I think, 3 or 4 different main companies that produce these particular repellents. And of course, they haven't been widely evaluated, unlike ITNs or IRS as well. And we found that at our 3 sites, the use of repellents was common. In Rourkela, nets are used a bit more. And we had a look at the percentage of malaria infections as well, and that's shown here. So basically, to the right-hand side is no use of repellent or vaporizer or coil. and then to the left-hand side is use of repellent or vaporizer or coil. So you can see that repellent use is associated with significantly less malaria. This is among our clinic study participants. Actually, in our survey participants, we didn't always find this. We didn't always find an association. And so we've really ended this study saying further clinical testing and evaluation of safety would be very useful for some sort of evidence-based recommendation of the use of these repellents, especially seeing as there's such a big industry in India. We also undertook a reactive case detection, and we found it wasn't a useful strategy at our sites. We did this in Nadiad and Rourkela. And just to briefly go through this, basically an index case would go back home and the household members would be surveyed, and then the neighbors as well, within a particular distance, and proximal and distal to that. And we did this in Chennai. Here's Chennai, and the index cases indicated, and then the roundabout surveying that we did, and then also in Nadiad too. So out of approximately equal number of index cases in Chennai, we only found 4 cases that were positive by PCR in close to 900 contacts. This is a very urban, densely populated area. No malaria in 131 contacts. There were enormous logistical difficulties for us to do this, just getting from A to B, the people being unwilling to participate, especially if they were leaving for school or weren't present during the day. Temperatures often exceeding 40 degrees Celsius and very labor intensive as well. So we've really, I think, ended this particular study saying we're not entirely sure about the value of reactive case detection, especially in a Vivax endemic setting. And especially in our intensely urban area. Okay, moving on to a second area, and this is looking at the environmental conditions in India determining malaria transmission with a number of different collaborators, including Matt Thomas here. And Matt in particular was very interested in trying to characterize the microclimate in urban settings. When you think about it, the temperatures that a mosquito has in a thatched building as opposed to in a concrete overhead tank must vary quite tremendously, and that's got to vary how the parasite develops in the mosquito. And sure enough, it does. So over about a year or so, there was a pilot study putting loggers into these different structures, which are shown down here, throughout Chennai. And you can really see the temperature differences here. This is the NOAA, so this is a local station, National Climatic Data Center station, and you can see how widely different actually the data loggers are to the reported NOAA. Another study we did was to try and identify where the immature mosquitoes were breeding. This, once again, is also in Chennai, shown here. So these are a lot of the cases from 2012 and from 2013 plotted. And we did a 12-month weekly study of the available clear and clean water and mosquito breeding habitats. And we found that overhead tanks really seemed to contribute significantly to the immature Anopheles stevensii burden, as shown down here. And that's just been accepted for publication. And we also found a preference of the adult stevensii for cattle sheds in Chennai too. So a big problem in Chennai is these illegal cattle sheds. They're indicated in blue, bright blue, shown here, kind of magenta color. And these are cases from 2012 and 2013, shown here. The problem with these illegal cattle sheds is that they really seem to be a place where Anopheles stevensii adults like to stay. You can see down here, for example, the figures, the numbers that we were able to collect from the cattle sheds compared to dwellings which are very close. All of the cattle sheds were right next to human dwellings within 5 meters or so. We also found the first report from Chennai of Plasmodium parasites in Anopheles subpictus as well, which is something that Pradeep had mentioned in his talk too. So just a few snippets then of our transmission studies. We've also been developing and using next generation genomics as well to study the genetics of malaria parasites. And this in particular is with Andrew Reed at Penn State. And we've developed a next-generation sequencing facility in Delhi. We decided to use the Ion Torrent PGM platform. This is it shown here. It's basically a benchtop sequencer, next-gen sequencer. You can take it out of the box. It's basically like a big pH machine, and it identifies hydrogen ions as the DNA strand extends. And this is the chip here. These chips where you put your sample and run on the machine. And we actually thought it would be the easiest system to have because it doesn't require a lot of computational power and bioinformatics. However, we do provide an enormous amount of bioinformatics and wet lab support. These are two of the postdocs in the lab who are basically on call all the time and provide constant support for this particular facility, and also through training as well. We also find it very difficult to get kits and reagents in India, so we have to purchase and ship a lot of kits from the US to India too. And some of the things that we're starting to develop then on this particular platform, we've developed an amplicon-seq method for surveillance. So this is a way that we can multiplex 96 samples and looking at 6 genes. So we can look at 600 total in one particular run. And we've developed this for a whole range of different drug resistance markers for falciparum, but also for COI markers, so markers which are very genetically polymorphic, and we know will tell us how many clones are in an individual too. So this is the schematic here. This is sort of the coverage. It's the whole length of the gene. It's not just a portion of it, which is wonderful for identifying new potential mutations as well, and also for identifying mixtures of resistant and sensitive clones within the same patient. In fact, we have one particular patient here, and you can see the mixtures of the alleles shown here for 3 different genes. These are some of the new mutations that we found in the K13 propeller, none of them associated with artemisinin resistance. Right now, we're running all of our samples through this amplicon-seq pipeline, basically so that we can track changes in the frequency of the parasite clones during the course of a drug treatment. So we've got day 0 and then follow-up for various patients, and which also should hopefully give us an indicator of drug resistance emergence too. One of the interesting findings over the years, by looking at a number of single copy genes or a handful of genes, is that Vivax seems to show greater genetic diversity than falciparum. And this is one of the topics that we wanted to investigate. And in particular, we wanted to generate the first Vivax and falciparum whole genomes from India. There were no whole genomes available when we started this work. And as Pradeep mentioned, that means that many of the papers up until now were unable to show what India genetic diversity looked like. So we rectified this. Here you can see a number of Vivax strains from Brazil, India, Mauritania, North Korea. And we chose 5 falciparum lines which matched from the same regions. We ran them through a next-generation sequencer, the Illumina, and had a look at the genetic diversity, and that's shown here. You can see in this particular figure, you've got the genotype quality along the left here, that these are all the falciparum isolates, these are all the Vivax isolates. Vivax really seems to show twice as much in this particular figure, genetic diversity compared to falciparum. And it doesn't matter whether you look at a different type of genetic markers such as, or type of genetic variations such as microsatellites shown here, or where you look in the genome too. And this genetic diversity has functional consequences as well. We found a lot more non-synonymous SNPs in red blood cell invasion and immune response genes as well. I'm not showing the data here. And this made us pronounce that actually vaccines targeting polymorphic antigens in Vivax may even be more compromised due to potential strain-specific immunity, which we already know occurs in falciparum. And so our last sentence of this particular paper, which was published in Nature Genetics in 2012, is that it reinforces the belief that Vivax malaria will be the more difficult parasite to eliminate. And I still very much believe that. Now I have a bit of a weird thing to do. This next set of data is under embargo at Nature Genetics, so I need to ask no live streaming, please. This is at the request of Nature Genetics, not me. So I apologize, otherwise I would show all of this. And it's a collaboration then with Sanjeev Mahanti, Dr. Satapathy, Terry Taylor, and then also Arian Dondorf, who actually was one of the key people who first started working with Sanjeev Mahanti and Saroj Mishra about a decade ago. And so we're taking advantage of 2 MRI platforms in endemic settings to look at cerebral malaria. Obviously, cerebral malaria, the pathophysiology is really not known at all. So this is Ispat General Hospital. It's close to 700-bed tertiary care hospital, 1.5 Tesla. For example, in 2015, we had 130 severe malaria patients, 53 with cerebral malaria, 11 pediatric as well. So we see a lot of adult cerebral malaria cases. This is Queen Elizabeth Central Hospital in Blantyre, Terry Taylor's institute. And they have a 0.35 Tesla. Since 2009, they've seen about 450 cerebral malaria cases, all of them actually children. So this was a special project then, very much facilitated by Mala as well, between these 2 particular isomers, the Malawi isomer, India isomer, running out of time. These are the MRI machines here. Our really interesting findings shown here is that brain lesions are identified in 50% of children and adults. So these are the number of cases that we've collected. These are the number of children here. And in fact, we have a very interesting finding that the swelling that's found is very redolent of a particular syndrome called posterior reversible encephalopathy syndrome. So you can see here this is a 5-year-old patient where there is a thickening of this entire cortical mantle, and then 48 hours later is completely resolved. And this is a particular syndrome which has been identified a few years ago. This is the first time there's been a link then between severe falciparum infection and PRES, this syndrome in non-fatal CM malaria, and that's just been submitted for publication. Okay, just wrapping up, this is a kind of fun slide. A few years ago at our SAG meeting, we had to put together the numbers, I think, so we could be quantitative about our success, and so this is a slide kind of left over from that. Number of household sensors, close to 5,000, individuals enrolled, 10,000, samples collected, PCRs, protein assays, I haven't talked about that. And then publications, front covers, a bit more fun. Number of retirements, 4. Number of weddings, 10. There are a lot of weddings in India. Number of divorces, 1. At least that's all I know of. Number of CSMI children, 7. A lot of babies. None in the USA. And we had a US Senate staff delegation as well. So this was the majority Senator Tom Harkin and a minority senator too, and they're directly in charge of the 170 billion DHHS budget. And so we took them around our Chennai field site a few years ago. So, okay. And one last thing, actually, sort of joking aside, there are 2 key people in our isomer that I really want to thank, because both of them passed away. Hema Joshi was instrumental in a lot of the genetics and genomics work that I'm involved in India. She was my main collaborator. And then Saroj Mishra, who passed away earlier this year, who was at Ispat General Hospital, who really started a lot of the MRI studies as well. And I'll leave it there.
We have time for 1 or 2 questions.
Very nice talk, Jane. My question is, like in India, I'm sure you know the history. In 1970s, the malaria is kind of almost eradicated. At least the government was, I don't know whether they announced it or not, the malaria was almost eradicated. So the Indian Malaria Research Institute, I think they do have a lot of parasites collected before 70 or mid 70s, I'm not sure. Do you look at the genotypic difference between the early 70s falciparum parasite genotype or the Vivax genotype and the one you collected now, the current one?
We haven't actually, but that's a really interesting point. Maybe we should look into that.
Any other questions? Okay, thank you.
Great, thanks.